I would like to introduce Dmytro Fishman on the subject, Seeing Deeper, Successful Industry Collaboration to Transforming Microscopy Image Analysis. Dmytro, please.
Thank you for the introduction. And indeed, today I wanted to talk about seeing deeper, or how can we use technology, specifically AI, to transform, to analyze microscopy and biomedical images. All right. So at first, when I have to explain the magnitude of the problem that we are dealing with as in biomedical image analysis researchers, I usually ask people to think about a very large number. Since people don't go too far in their thinking of the large numbers, I prime them to think of a specific large number, and specifically number of stars in the galaxy. And this is about 200 billion stars. When it turns out that this is not enough to explain really the complexity that we are dealing with as researchers, because the average human body has approximately 37 trillion cells. And that means that if you think about this a little bit, then the star is just a massive collection of more or less uniform matter, while human body is full of very intricate matter that communicates in very sophisticated ways. Okay, so basically our system, or human system, consists of huge number of cells that constitute tissues that in their own turn constitute organs that help all this system to function. And as very complex system, we can break in many different ways. One way how this can happen is that one rogue cell acquires the potential for dividing uncontrollably, invading its own tissue, later suppressing the organ, and finally metastasizing into other organs, killing the host. So at this point, we come in as researchers in biomedical image analysis, and we as a community, on the other hand, we were very successful so far in solving different types of problems. We have been very good at classifying lots of images into a large number of classes. We have been building systems that can navigate, that can control cars with very little human intervention. There was research mimicking human artists with the very high fidelity images that are being produced. And you might ask yourself a question: how these problems on the left side of the slide correlate, and how they help to solve the problems on the right hand about the cancer? And it turns out that there are a lot of problems that happen with the humans that we can formulate in the same image type of representation that we know how to solve, and we have the methods to solve. I'll give you a couple of examples. For example, we are working with microscopy images that look like this, and they come from a company called Revvity. This is the cell line, cancer cell line. So these images are images of cancer cells. And when biologists, they work with a new drug, they want to test it on cells first. So they apply this new drug to a cell line like this. And then they want to know how much the cell count has changed, how many cells died, how many cells have shrunk, etc., etc. So we help them to do just that by applying deep neural networks, such as this one, to detect cells and quantify cells. So not only that we count cells that we find, but we also tell a little bit more detail, as in how the cells look like, what's their shape features, what's their size features, etc., etc. Not only this type of images that we work with, but we also work with a different type of images, like this. And they're called bright field images. And the bright field images are a lot more complex than the previous images that I've showed you, fluorescent images. And the reason why is that they're very low on contrast. It's very hard to see the cells on these images, albeit it is possible. It's very hard to really analyze these images by a human eye. So it's no longer possible to do a manual analysis. We have to do something else. And it turns out that very much the same models that we have been using can detect cells even from these challenging images. So this is what we do. We essentially detect and count and characterize cells from the bright field images. And the interesting thing is that this kind of system has already made its way into a production of the company, Revvity company, the ones that we've been collaborating with all these years. And they are now offering this model that we have developed as part of their offering in for their very wide network of clients. Moving on in the letter of biomedical images that I've showed, the next step is the tissue analysis. So at this point, a lot of pathology labs around the world work with images of histopathology that you can see here. These are usually pieces of tumor tissue extracted from the patient through a procedure called biopsy that you can also see in the slide. And then what happens is that pathologists in those labs, they are manually looking through these images and doing all sorts of studies. They have to quantify all sorts of items. For example, they have to really count the number of cells on those images. They have to find and measure the volume and the area of certain structures. For example, this is a human testis, and what is of interest is the size and the shape of the tubuli. And this is what we help them to, this is what we help to automate. So we are building models that can detect those structures, segment those structures, meaning measure the structures that we detect, and also quantify, let's say, cells from those histopathology images. And at this point, we're working with Estonian hospitals, with Ida-Tallinna Keskhaigla and also with Tartu University Hospital to actually bring these models into practice of the pathology labs and automate this manual work that currently consumes a lot of man hours without much really need to do so. Finally, moving on to the final step in the letter is organ level analysis. We're working with a company called Better Medicine that I'm a co-founder of to detect and measure and report malignant lesions, basically tumors, from the CT scans. So you see a CT scan here on the image, and what our models help to do is they help to really annotate, pre-analyze those images for the radiologist. Before the radiologists step in, they help to figure out where the different lesions are, how dangerous they are potentially for the human, and they provide all sorts of relevant measurements. So they measure the volume, they measure the surface area, they provide a written report that radiologists could use instead of writing by themselves manually. And this also has been shown to save a lot of time from radiology workday and help them to focus on more challenging and more complicated tasks instead. Just to kind of summarize a little bit here is that we start with the lowest level for the human that would make sense for a human. We start with cells, where we work with companies such as Revvity and Syngenta to analyze, to segment, to detect cells from various types of microscopy images. So bright field and fluorescence were just examples, but we did work with many others. And this helps to automate a lot of processes in drug discovery experiments, in human, basically in human analysis and human studies experiments. Secondly, we move on to the tissue level, where we also help to automate a lot of manual work currently done in pathology labs, where people were supposed to manually detect, segment, and measure all sorts of structures in the histopathology images. We help them to build models that automate such workflows, making them more productive, making them more efficient, and maybe even accurate as well. Finally, on the organ level, we build models that can detect lesions, that can detect tumors, that help radiologists to measure, to detect, and report these lesions as findings in their reports. Again, helping them to save time, helping them to be more productive and also more accurate, because models do not require a lot of rest. They are just automata working in the computer, while doctors do need rest and they do need to attend to more complicated and challenging cases. And this is a short summary. This is where we help with our AI models. This is just a small history slide here. This was the very first hackathon meeting, because my talk is entitled as a successful collaboration. So the successful collaboration meant in the talk was collaboration with a company called Revvity. This is where we started. This is where the whole process began. We started a long time ago, so 2016, with the very first hackathon, which was organized between the Institute of Computer Science and the company. And we sat together for a few days to try to solve a very challenging at that time problem. And the results were very convincing and very promising. That's why we started this collaboration this many years ago. And since then, we have been every year delivering all sorts of systems and helping them to automate a lot of processes inside the company. Finally, this is the research group that does all this work. They're called Biomedical Computer Vision. There are a lot of students, also a lot of there are PhD students, there are master students, there are bachelor students who work on these problems, who help us to bring AI into the industrial world and industrial setting, basically solving all these challenging problems that I've described. That's all from my side, so thank you very much.
Yes, thank you, Dmytro. We are sure to have some questions for you, so please stay here. Okay. Because it's, well, quite literally life-changing technology that you were talking about. And perhaps one of the first things I'd like to ask you about is if it's something that can actually be used to help people. How quickly do these collaboration projects bear fruits? Is it like we need to spend a whole lot of time in R&D, we need to spend a whole lot of time doing like clinical trials, so actually the collaboration project takes a lot of time, you know, for the university, for the scientists themselves, or is it something that you can be reasonably confident that you can already give to existing hospitals or private companies as a product?
So I think there are two important parts of the question. One is about the product and the other one is about the speed of the research. And I think when we talk about the research, it has become a lot faster because we gained a lot of experience. So now, for example, we are talking about this histopathology image analysis to the local hospitals. And what happens is that they sometimes require additional system to be built to automate a new process, a new diagnosis that they're making. And this does not take too much time. What does take time is acquiring enough data for these models to work and annotating that data. So this is what I think I would say majority of the time. The rest of the technology behind it, we have enough experience to plug some of the things in and get the results relatively quickly. Then, of course, there is still a lot of time that comes with just fine-tuning these results to make them more efficient, to make them a bit more accurate, etc. So this process does take time, but getting very first proof of concept results is very fast. So it's a matter of weeks rather than months. Regarding the product, especially in medical setting, this takes a lot of time because of the certification programs. So you can't really develop a product and go ahead and use it in the medical practice right away. So you need to certify it, you need to show that it's safe, you need to run maybe a clinical study where you confirm that it makes sense. But obviously there are use for research, so you can use some of these things for the research purposes. There you don't have to provide a lot of certification, yet you need to have an ethics committee application approved for this specific use case. So in short, research part does not take too much time to the proof of concept stage. Product does take time because of the relevant regulations.
Okay, I see. So not that kind of a fast food approach that a company can come to the university and say that give us something instantly, but
no, I think we can give something instantly in a way that we can show them that the promise is there, that this will work or this may not work. So we can answer this question relatively quickly. Obviously, getting to the product is what takes a lot of time, but also it requires a lot of attention from the company's side because research is not the product. So we can't really give them the product. So product requires also company on board. So we are kind of helping with the first part, but they have to take it on and work on the product for themselves.
That's an interesting thing that you raise is that the research is not the product, which was something I was thinking about if a company were to approach you and they were not interested in specific application that you are designing right now, but just in your general know-how, your general expertise, and they were to present you a different kind of project, which might not be specifically like microscopy analysis, but would have something to do with different images to use AI on and try to get some kind of results from that. Would that be something that could spark your interest?
Yeah. I mean, certainly. There are very interesting application areas where we really would like to step in. So totally, we are interested in experimenting with new data and new industries and new fields, and this is all very interesting. That's how we started to work with Syngenta, which is an agricultural company which is working with fungi, images, etc. So there are a lot of variability in the field. It's a lot about images, so it's all about these kind of things. So there are a lot of companies working with this stuff.
Similar to that, so if there's a lot of companies working on this kind of stuff, what exactly gives you the edge? Something to make people think that you are at the bleeding edge of this kind of approach?
I mean, we are. I think as many other research groups, all of us are forced to be at the edge because if we are not at the edge, we are not able to provide the most efficient solution that's going to get this competitive advantage, right? So the competitive advantage is what matters for the companies, and competitive advantage is you can only get it through the using state-of-the-art approaches. So there is no way you can get the state-of-the-art without really being at the edge. That's the strength. Being a research group, working with the current literature, implementing new approaches is what gives us the edge, after all, because we are not bound to the old technologies. We can experiment. We have the freedom to experiment, and that's what helps us a lot.
I wasn't trying to interrupt you.
Yes.
Yeah. That's the other question I had about experimentation in that regard. If you have like your partner is basically your own company, like you said before, Better Medicine. Is that something that gives you more kind of self-assuredness or an idea that you have this a little bit more room to experiment with your approaches, that you don't have to give your results immediately to some kind of possibly anonymous firm elsewhere?
I think it's not about having my own company as part of the project, what really gives that feeling, but more of expectation management bit. When the companies approach, we always try to maintain the expectation that this is research. And what it means for the company is that it can go both ways. It can result in great technology that they can use for their competitive advantage and win the market, but it can also prove to be wrong, and it can prove to be useless, and maybe it's not going to work. So we need to be very transparent about this and communicate very openly about potential risks of investing into the research. It's a high-risk investment, but it's also a potentially high-reward investment, because if it does work, it is a competitive advantage for some time, right? So the ability to grasp more market. So in that sense, I think startups are not, otherwise if it's not just for my own company, but startups are not that kind of willing to do that kind of investment, but more of well-established companies. They understand the value of the research, they understand the risks, and they have enough capacity to invest into high-risk type of things, like, for example, Revvity does.
Mm-hmm. And possibly give you the resources and the time you need to actually develop it.
And not to pressure too much, and be patient, and also guide us a little bit, we have been extremely lucky with the collaboration.
But how do you think you can manage these kinds of expectations? Because if a company does approach you and they have these expectations to obviously to have some kind of return on investments, what is the way to be possibly firm and transparent from the get-go?
Yeah, I did a lot of strategies how we manage these expectations. We communicate very frequently. So we are keeping the company completely inside the processes. So we are telling them everything we do. We are willing to explain every other part of the research. So this transparency helps to build the trust, but also it helps to build this kind of realistic timelines. So we're just telling them that this is what exactly we do. If you guys know how to do it faster, come and talk to me.
Yeah, everyone would like to hear that, including you.
Right.
As the final question, in this kind of industry collaboration, how much do you learn from the other side?
Oh, a lot, so much. So again, Revvity example, Revvity is specializing in microscopes. This means a lot of optical research, a lot of know-how about how the light travels through the different medium, water, air, what kind of images you get, and there are differences, and there are differences in terms of what kind of microscope is used. All of that comes from the company. So a lot of domain knowledge comes from the company. And they know their user base, they know their client base, they know what exactly kind of interesting stuff they want to research and what kind of stuff is maybe not so cool for them. So all of that is, it's not one-way communication, it's always two ways.
In a way, it's like this, if I may say so, this kind of boring practical stuff that you might not think in the high field of research, but it is necessary.
Becomes so important at the end of the day. And also we learn so much ourselves. So yeah, both ways.
Well, that's very great to hear that industry collaboration goes both ways. But thank you very much, Dmytro Fishman, lecturer at the Institute of Computer Science.
Lecturer of AI. Thank you.
Lecturer of AI. Very good, thank you.